Cell division, an essential life process. What happens to the cells of injured tissue? That injured tissue gets cured. That means new cells are formed in place of those injured tissue. So in healing of wound, new cells are formed. Do the plant get injured when we pluck the flowers? Yes. But that cut is then healed after some time by formation of new cells. Then during the growth of any individual, new cells are formed. How the new individual of a species is formed from existing one of same species? By the process of reproduction. And this reproduction can be sexual or asexual. So cell theory tells us that new cells are always formed from the pre-existing cells. So the process of formation of cells is continuously going on in case of reproduction, in case of growth, in case of wound healing. So cell division is one of the very important life processes in cells and living organisms. Due to this property only, a new organism can be formed from existing one, that is reproduction, and a multicellular organism grows up. An emaciated body, that means injured body, is healed, is restored. There are two types of cell division, namely mitosis and meiosis. Mitosis occur in somatic cells. What do you mean by somatic cells? Somatic cells are cells in our organs other than the reproductive cells. So all the cells in your muscles, in your digestive system, in your transportation system, they are somatic cells. And there are stem cells in the body. Stem cells are the cells which can be differentiated into new kind of cells. So in somatic cell and stem cells, the process of mitosis occurs in cell division, whereas meiosis occurs in germ cell, that is reproductive cell. So before we study the cell division, we should know the structural organization of cell, and we know that a cell has a nucleus surrounded by cytoplasm, and there are many cell organelles in the cytoplasm. Let us study the cell division with the help of all this information. So before any type of cell division, cell doubles up its chromosome number. Now we know that every organism has 2 into n number of chromosomes in its body, and from where this 2 into n number comes, n chromosome come from the father and n chromosome come from the mother when the zygote is formed for that individual. So these 2n chromosomes are present in each cell. Now these 2n chromosomes are first converted into 4n chromosome before the cell division. So the chromosome number is doubled up by formation of sister chromatid, as we will see afterwards. So first remember, in mitosis, the first step is the 2n chromosomes are doubled up to 4n chromosomes. A pair of each chromosome is present in a 2n condition. And as we have said earlier, one chromosome from father, one chromosome from mother, and then the pair of chromosome is there. Whereas single chromosome of each type is present in n condition, and their structure is like as shown in the figure. So mitosis occurs in somatic cells and stem cells. Mitosis is completed in two main steps. One is karyokinesis, that means the nuclear division, and the next step is cytokinesis, that is cytoplasmic division. Karyokinesis is completed through 4 steps. So first we have mitosis. Mitosis has 2 steps: karyokinesis, nuclear division, and cytokinesis, cytoplasmic division. Then karyokinesis is further divided into 4 steps, and the first among them is prophase. In prophase, the condensation of thin thread-like chromosome starts. That means when cell is not dividing, the chromosomes are in the form of very thin threads which are invisible through the microscope. But in the prophase of the cell division, the condensation of thread-like chromosomes start. So in this phase, chromosome becomes short and thick. They start to appear along with their sister chromatids. So that we have discussed that 2n chromosomes will be converted into 4n chromosomes here, isn't it? So here you can see the 2n chromosome, and you can see the condensation of the chromosome taking place, and after the condensation you can see the formation of sister chromatid here. So now the 2n chromosomes are converted into 4n chromosome. Centrioles duplicate, and each centriole moves to the opposite side of the cell. You can see the centrioles here. So centrioles are duplicated, and each centriole goes to the opposite end of the cells. Nuclear membrane and nucleolus start to disappear. So this is the prophase. That means here cell is preparing itself to divide the chromosome. For that, what will happen? First, the chromosome will become short and thick. Then they will be replicated. 2n chromosomes will be converted into 4n chromosomes. Then centrioles will duplicate, and they will move to the opposite poles of the cell. Nuclear membrane and nucleolus will start to disappear. Next is metaphase. In the metaphase, nuclear membrane completely disappears. Chromosomes complete their condensation and are clearly visible along with their sister chromatids. In this phase, that is the metaphase, chromosomes arrange themselves along the equatorial plane. So you can see here the chromosomes, they are arranging themselves like this, parallel to each other in the equatorial plane, central plane. And you can see the special type of fiber is formed between the centromere of each chromosome and both centrioles. Centromere means this point. Here you can see the centromeres. So between the centromeres of the chromosome and the centrioles at the pole, flexible protein fibers, and they are called spindle fibers, they are formed. So this is the metaphase. In anaphase, centromere split. That means the chromosomes are separated and they are pulled by the spindle fibers to the opposite end of the cell. So in the anaphase, centromere split and sister chromatids of each chromosome separate and are pulled apart in opposite direction with the help of spindle fibers. Separated sister chromatids are called as daughter chromosomes. So now we have again 2n chromosomes. Are you understanding? First we had 2n chromosome. After the formation of sister chromatid, they become 4n. Now the 4n are again divided into 2n. So now the chromosomes have been duplicated. Chromosome being pulled appear like bunch of bananas. In this way, each set of chromosome reach to the opposite poles of the cell. So here you can see they are pulled and they are going to the opposite end of the cell. Finally, the telophase. Now in telophase, the condition of the prophase reappears, but now we have 2 nuclei. So the chromosome which have reached to the opposite pole now start to decondense as they were in the prophase, due to which they again become thread-like thin, invisible. Nuclear membrane is formed around each set of chromosome reached at poles. Thus, 2 daughter nuclei are formed in a cell. Nucleolus reappears, spindle fiber completely disappear. So this is the telophase, and in this way the karyokinesis is completed. The karyokinesis has been completed, and now the cytokinesis begins. Cytokinesis takes place in 2 different ways in animal cells and plant cells. In animal cells, a notch is formed at the equatorial plane of the cell, which deepens gradually, and thereby 2 new cells are formed. In case of plant cells, a cell plate is formed exactly in the midline, and the cytokinesis is completed. So in this way, the karyokinesis and cytokinesis is completed. This is mitosis. Mitosis takes place in case of somatic and stem cells, then during the growth of the body, then healing the wounds of the body. It has 2 steps, karyokinesis and cytokinesis. In karyokinesis, we have prophase, we have metaphase, we have anaphase, and the telophase. Whereas in cytokinesis, a notch is formed at the equatorial plane. In animal cells, cell plate is formed along the midline in the plant cells. So mitosis is essential for the growth of the body, restoration of emaciated body, wound healing, formation of blood cell, etc. For growth, restoration, wound healing, and formation of blood cells. Now the next type of cell division is meiosis. Meiosis is completed through 2 stages, and they are meiosis 1 and meiosis 2. In meiosis 1, recombination crossing over occurs between homologous chromosomes, and thereafter those homologous chromosomes, remember homologous chromosome, they are not sister chromatid, are divided into 2 groups, and thus 2 haploid cells are formed. So this is very interesting. Let us see why does this happen. Now in case of reproductive cells, when the fusion of male and female gamete will occur, number of chromosome will be doubled. So initially in that reproductive cell, we should have half the number of chromosome. So from 2n chromosome, we want cells having haploid, that is n chromosome. So how does this happen? We know that each chromosome is in the form of a pair, correct? So if suppose first chromosome, the 1 chromosome will come from father and 1 chromosome will come from mother. Now for these 2 chromosomes, recombination and crossing over will occur, and from these 2 chromosomes, a single chromosome is formed, which is neither like this chromosome nor like this chromosome, but it is the combination of the two chromosomes. So crossing over occurs here. That means some part of the father chromosome and some part of the mother chromosome is mixed in the formation of new chromosome. And every time this recombination, crossing over, takes place in a different way. And that's why each reproductive cell, each sperm cell, each egg cell is unique in nature. It has got unique chromosome combinations. So in meiosis 1, the main thing is recombination, crossing over, and hence two haploid cells are formed in meiosis 1. In meiosis 1, two haploid cells are formed. Means two cells having N chromosomes are formed. And these N chromosomes are different. They are formed by recombination, crossing over. Now meiosis 2 is just like mitosis. But this is mitosis of N chromosomes now. Correct. So it has got prophase 2, metaphase 2, anaphase 2, telophase 2, like that. Meiosis is just like this. Meiosis 2 is just like mitosis. In this type, two haploid daughter cell formed in meiosis 1 undergo division by separation of recombined sister chromatids, and four haploid cells are formed. Four haploid daughter cells are formed. Process of gamete formation and spore formation occur by meiosis. Formation of gamete, that means reproductive cells in sexual reproduction, and spore formation, asexual reproduction, occur by meiosis. In this type of cell division, four haploid daughter cells are formed from one diploid cell. So in all we have 2n cell, and from that 2n cell, four haploid cells are formed. And each cell is unique in its chromosomal structure. Due to this, all the four daughter cells are genetically different from parent cell and from each other also. And that is the reason of diversity we found in sexual reproduction. Sexual reproduction happens in human beings, so you can see even the brothers and sisters, they do not look exactly same. They look different. Even though they have inherited their chromosome from same parents, due to this recombination and crossing over, each one of them has a unique set of chromosomes. Only the like twins, they have same DNA, same chromosome. We'll see next chapter how like twins are born. Take a medium sized onion, keep it in a conical flask with water, wait for 4-5 days, cut the tips of the roots, then put some drops of iodine, that is the coloring material. Then take one of the root tip on a glass slide, cover it with a cover slip, add 1-2 drop of water and observe it under microscope. You will see the cell division in all those cells. So what do you mean by diploid cell? Diploid cell is the cell having the pairs of chromosome. The cells, all somatic cells, stem cells, they have 2n. That means they have pairs of chromosome. But from these cells, when the reproductive cells are formed, they are haploid. That means they have only half number of chromosomes. Homologous chromosomes are the chromosomes in the pair of chromosome, out of which one chromosome is inherited from father and other from the mother. Whether the gametes are haploid or diploid, gametes are haploid. They have half number of chromosomes. However, haploid cells are formed. Haploid cells are formed by the process of meiosis. What is the importance of the haploid cell? Only due to the haploid cell that after fertilization, number of chromosome is restored. If the cells were diploid, then the next generation number of chromosome will become double, and that is not wanted. So, due to haploid cell, number of chromosome in the next generation is restored.